Can you hear me at the back? Okay, great. Thank you so much for having me here, and what a great audience. I guess New York is big after all, right? So it's going to be quite a different style, which is great, right? Let's change it up a little bit. If anyone's seen my talks before, they move quite rapidly through a lot of information, so hopefully get it done in enough time. To avoid getting missiness at the end, I just wanted to say that again, like these things are not done independently. I count on great collaborators, particularly in Dr. Alison Roy, the USGS co-op unit at UMass Amherst, as well as Michelle Stogginger in the Climate Science Center at UMass Amherst. Those are two of my key collaborators when it comes to river herring, and I really wouldn't be able to do it all alone. So, and then there's just a litany of grad students that we both current and ongoing that are working on these questions, and again, that they do the bulk of the work, and I put up a slide that takes them 2 years to make and take all the credit for it. Okay, so we've had a bit of an introduction to alewife. In coastal systems, there are the two species, alewife and blueback herring. They often migrate slightly different timing because blueback seem to prefer a little bit warmer temperature. So, but they both school, and they're pelagic and forage fish for a large number of predators, and they also both graze on essentially the same kind of zooplankton. Because they move around a lot, they have a large effect on nutrient transport, as well as they're just this amazing cultural moment, which John talks nicely about in Running Silver. And it's just this thing to see on the coastal systems as these river herring return every spring. And I'll show you a little bit of that in a second. They're anadromous, repeat spawning fish, so they'll return, as long as they're still alive, they'll return year after year after they reach maturity. So, just wanted to mention, I think it was covered really nicely by Brian, but there's alewife in particular, and I honestly don't understand why we don't have landlocked blueback population. I don't hear anybody talking about that. But alewife, Sarah Turner at Mass DMF always says they're like rats. They'll just like spawn anywhere, and it actually seems to be kind of the case. We see spawning in rivers of both species. We see both species hybridizing in lakes quite frequently. We see pure bluebacks in lakes. We see both species spawning in sort of tidal areas, and so they just seem to go wherever they can possibly get there. But because of that, they will play different roles. But for today, I'm just going to talk about these anadromous runs. I'm not going to talk too much about the landlocked populations because they were just covered nicely. So here's an anadromous life history. We start in the ocean, and they spend the majority of their lives in the ocean, up to 3 to 4 years, before they start returning to spawn. They make their spawning run. They lay these demersal eggs. And those hatch after about 9 days in Massachusetts springtime temperatures. The larvae emerge, they start feeding about 3 to 5 days post-hatch, grow up to be these juveniles, and then sometime between June and the next spring, depending on water flow and so forth, they'll migrate out into the ocean, where they'll complete their growth and return a number of years later. There's these moments that happen. It sounded like it died there for a second. All right, so there's these moments that you can see where they migrate out, migrate into rivers, where they spawn and hatching, that if we knew something about what happened at each of those moments, we would have a lot better understanding of their life histories and where there are issues along the Atlantic coast in terms of the population's recoveries. And the reason for that is these are essentially kind of black boxes right now. I always hear people talk in the ocean, who work on the ocean, call the freshwater part the black box, and the people who work on the freshwater call the ocean the black box. I think they're both black boxes, and if you want an analogy for a black box, it's sort of like if you're looking in a program and you look for someone's abstract and it's missing, and you always see the title. That's sort of like the level of black box we have here. So we sort of know where they are, but not what they're actually doing in many of these systems. And this really impedes our understanding of their populations. So we have this ocean migration into fresh water in the spring. And this is where they choke into these little areas. They're counted fairly accurately through a variety of different mechanisms, both citizen science, electronic counters, video counters. And that's the information we have. It drives almost all of our management reaction to their population. We do have some trawl surveys, although now I'm going to think about looking at those again a little bit, but they tend to be in the spring and the fall, and they don't really seem to reflect what fishermen are catching or were catching and the decline. So it's really these counts that are important. So here's what a count looks like from a year. This is a snippet-to-it system on the coast of Massachusetts. You tend to get these sort of peaks up and down from day to day. I'll talk a little bit about why later on in the talk. Sometimes you get a second bit of a run, and that's off. Sometimes bluebacks. Other times it's just because it got really cold right here, or there was a big rain event that impeded passage through a fish passage system. All right, this is boring data. So we'll just look at this for a second, just to give you a sense of what's going on. This is not a particularly large run. This is actually in the Parker River, and it's during the day, and this is just opportunistic. There's some sound effects too. It's a really chaotic scene, and so you can imagine counting this being a challenge. Also, if you just see, there's some eels, little glass eels flying around as well. So it's a really fantastic thing to witness, and so I just encourage it. If you ever get a chance to go and see it live, do so. Okay, so then we take that data that we collect from that, and we use it for things like this, which is just sort of looking at the population trend over time. Here's that decline that really happened in the late 90s, in the 1990s through the early 2000s. And then this really frustrated me. These are a series of stock recruitment relationships that were a big part of the early stock assessments for these Atlantic populations. And what you see is that there's the spawners, all right, so you get that, that's that count. Well, the recruits are often the same data. They're the data of the counts of the fish returning after 3 years in the oceans, and sort of estimating how many of those are first-time spawners. And that's your recruits. Which means that you don't know anything that happened from the time that they went into the lake, laid eggs, and until they came back 3 years later. And so understanding where the mortality or where there are issues in their recovery really impeded by that lack of knowledge. And so that has really set up sort of the black boxes that I've tried to sort of focus on a little bit in my time since starting at UMass in 2012. So what does this mean? So these are standardized counts now throughout. These are the longest time series, including the Connecticut River. So maybe there's a little pickup and down. I mean, it's really very difficult to really understand what's going on from this data. So I'm going to start the real part of my talk by first of all giving some historical context as to what we're counting there, and then I'll move on into the second half of my talk into sort of a data dump of information that we're collecting that we are hoping are going to feed into a better understanding overall of these populations. So first I'm going to talk about some historical catch information to try to understand past dynamics. Then some historical dam work that we've done to calculate lost habitat, all with trying to answer questions about the current and past ecosystem roles of these species, and also where we're going. Like, what do we want to have in the future to evaluate trade-offs and so forth in terms of fisheries. So the first paper I'm going to talk about is this work that Emily Klein did for her PhD at the University of New Hampshire. And what she did is got these really great data sets from the Bay of Fundy from the 1873 to 1920 period and the 1967 to 2014. She used a series of different, two different time series analysis, typical autoregressive technique, and then this more nonlinear dynamics that George Sukahara often champions as a way to detect nonlinear trends in data. Well, it's great for the historical data. You got these. And what she did is created time series for each of these locations, and then tried to evaluate each of these locations' time series. So what she saw is that if you get these nice time series for multiple locations for Gaspereau is river herring in Canada, all of these fish are in a high enough abundance and in enough locations to create these time series of their dynamics. They're sort of an aside for this talk, but they were actually quite predictable in terms of what direction they were headed, down actually in many cases, and there was also a nonlinear signal. But this was what I wanted to bring up for today. There's not enough data to even evaluate all of these species in these systems. And so it's just this complete change in their abundance and relative value for both economic value as well as ecological value. At the same time, we were also working on another paper with Karen Alexander as a lead looking at effects of a volcano and all kinds of interesting stuff. But I just want to show this figure to demonstrate that there was a time when anadromous fish in coastal systems, and this is using fish inspector reports, and basically what Massachusetts did at the time were like, there were these areas that were not producing quality fish, and so they sent out inspectors to sort of make sure that there was a high quality product that was being exported so that there would be a good, reputation and therefore would sell more product. So they collected this information. It's the earliest data that we have, so it's export data. There's a little bit of a bias there, but certainly what we can say is that there was a time when anadromous fish were equal in value in terms of export to these pelagic marine fish like herring and others, and that just totally changed. And something happens between here, the story in the papers about volcanoes and then the effect of dams, but I'll just keep it there for the purpose of this talk. So it's really not particularly important in these systems as we move forward in terms of an economic driver. Well, no wonder. I mean, this is what we used to see on our Atlantic rivers, where these enormous runs of fish that were capable of providing sustenance for locals, but also for export as salted or pickled products. Carolyn Hall, a student I helped advise at Stony Brook University, we published this work back in 2012, used that same fish export data for the first part of this data set. Actually, this is when we first collected and compiled this data. And then it gets more into fish fish commissioner reports, as well as some more contemporary data that's actually held by the towns. Towns hold the data of alewife harvest in Maine. And what it showed is that for this little period of time, there were really only these 3 river systems that were producing any measurable number of fish that could be exported. And what you determine, you can see this large white here, that's the Penobscot watershed. I mean, the Penobscot watershed should be producing a lot of fish that are capable of being marketed, but they're just not. So everything gets sort of pushed to the coast, and this is what started leading us to asking questions about the impact of dams. At the time, I was driving around Maine all the time, and I just you can't avoid noticing how many dams there are as you do these drives. So Carolyn compiled these data sets where she took these red are existing dams, the purple are actually historical dams that in many cases you can't even tell they were there. They're just they were abandoned back often in the Depression period or in other time periods where it just wasn't economically viable to do run a mill. And so they're just left in disrepair. Then there's the head of tide dams, which are when in Maine you can put a dam almost up to to where the tides are because you get an elevation change right up to the water level to the ocean. And then there's the upstream boundary. And what Carolyn did is just looked at how much habitat was being lost over time. And what she found, and this is sort of the money figure from all of the work that she did, is that if you look at in particular the amount of lake remaining, you just get this very small amount of lake that's remaining that could be providing habitat for these alewife and river herring. Then what we did is we took some estimates of productivity from that same data that I just complained about with the number of three-year-olds or estimated number of first-time spawners as recruits, and looked at like what are we missing by having this. So it basically tracks the increase in numbers of dams over time, but we're leaving some very abstract number of 3 × 10⁷ kilograms of fish that we're just missing from our systems and from our harvest, potential harvest. All right, so what does that number mean? Well, first of all, I'll point out that this is sort of the time period of where we had that fish inspector report. So even at the time that we started having this export data collected, these systems had already experienced a significant amount of habitat loss, over 50%. So just always bear that in mind, that no matter even if we think we have an early time series, that we still aren't capturing the early dynamics. This figure is the entire U.S. landings for both species, river herring, alewife and blueback, in the entire coast. And so the 9 watersheds in Maine were producing about what you see in the entire landings of today, of when things were good back in the 1950s, and then of course things have gone south since then. So we're really just not even on a ballpark of what these numbers should be. So I had another graduate student, Stephen Maddox, come in when I started at University of Massachusetts, and what he did was extend this by first taking the distribution of shad. There's a really good, interesting information contained in those U.S. Fishing Commissioner reports. I highly encourage young folks to spend some time learning about the history of these fishes, including stocking, by the way. It's incredible what we stocked and where. And what he did was then compile the same kind of information, this loss of lake habitat over time in all these different watersheds. This is the work that Carolyn did. This is the work that Stephen added. And what you see is that dams started in the areas that we would expect around the major cities as they started expanding and expanded inland and up the coast of Maine. There was some socio-political reasons why we didn't dam really early up in down east Maine. Again, just emphasizing the degree of loss of these habitats. So the one place that did seem to have quite a lot of habitat left was coastal Massachusetts, and actually it sort of reflects in the southern New England, seems to have a little bit more habitat remaining than in the Gulf of Maine proper. And we think that the real reason for that is because of the shape of the land. As you're up in this part, you can see that there's just a lot of elevation change pretty close to the coast. And so it's really easy to put a dam right up to that head of tide. Whereas in coastal Massachusetts, it's just a lot of really flat land down in the area around Cape Cod, a lot of marsh habitat in the Great Marsh, probably just sustaining the amount of habitat a little bit more. There are cranberry bogs though, and so that's a story for another day. Okay, so then Stephen tried to go one step further and again produce, and the reason why these are in these odd-looking numbers is exactly what Brian mentioned, is that we wanted to feed these into mass balanced ecosystem models, which I'll come to in a second. So instead of producing and giving you those numbers, I'm going to give you these numbers. So we're just losing a lot of forage. So whether it's these 30 billion little tiny fish that are in freshwater ponds, or these millions of fish that are out in the ocean available for oceanic predators, a lot of marine-derived nutrients. But this is one of the ones that I just like to focus on because 18 million additional fish is a lot of fish by our standards of today. And these are extremely conservative numbers. We used estimates from our freshwater productivity using our pelagic pursanes, which I'll come to in a little bit. And we know, and we use the median, not the average. Everything was to be conservative, and I think that these numbers do not really reflect the true capacity that we've lost in these systems. Well, river herring move though, and they move a lot, and so they open themselves to predation from a lot of different potential sources. And so, and the theme of this, I should have got a nice image. It's the sort of more muddled one off the website. But in the theme of the conference, I'm going to switch a little bit into the role that I think that this change has occurred, but also their current role even in ecosystem dynamics. I'm going to start in the freshwater black box. So just to go back to this, these dots here are predator ponds. These are systems where we did sampling for predators. And what Stephen did is took those predators and with a team of undergraduates, dissected all the guts out, measured all the fish, did all the things you need to do, and sort of looked at the incidence of alewives in the stomachs of these predatory fish. I did not expect brown bullhead to be the #1 fish consuming river herring, but they are. And in fact, one of the things you notice when you're out on these lakes in the nighttime, and I'm going to talk a little bit about our sampling later, is you see fish right at the surface, like feeding on these juveniles. We've seen eels at the surface. We've seen brown bullheads. We've seen a lot of fish just right there. Another big predator is white perch. We see those all the time, right in these schools of river herring. These are not eating too much in this study, but that's because we did this sampling later in the year, and so the fish had already achieved about 40 millimeters in size. And so we weren't, but what we do know from stable isotopes that there's definitely some consumption of eggs or very small larvae by these bluegills and pumpkin seeds. So a lot of things are eating them. Stephen then did for his second chapter of his master's thesis the following year, did a paired lake study where he found a lake that didn't have any alewives in it next to a lake that we had been doing sampling in for a couple of years, looking at the number of, looking at the productivity of juveniles. And what he did was just look at predators in each of these systems. I warned him not to use gill nets. I said, you're going to have nothing recognizable in their stomachs, but he wanted to use gill nets. So he used gill nets and didn't have anything recognizable in the stomachs. But he does have some interesting stable isotope data, which I'm not going to cover today, but sort of only reflects that everything eats them. What we did see is, if you look at the Fulton's K relative weight or hepatostomatic index, either in alewife density or in presence or absence of alewives, there's a big June boost to this is white perch, but also to yellow perch. A big boost in their condition, no matter how you look at it. And it seems to go away later on, but this is a lot because of some of the systems that he sampled really low densities of river herring later in the year. Then he took otoliths. This is a white perch otolith that he used the crack and burn technique to look at the ages and estimate the length and age relationship for these fish, and produce these figures, which demonstrated number one, these fish seem to be, because they get this boost of energy, they seem to grow a little bit faster early in their lives, but they, because of that, seem to actually mature a year earlier than they do in non-alewife systems. They actually end up smaller, interestingly, than they do in an alewife dominant, in a system with a lot of river herring in it. And this work's sort of ongoing, and Stephen's struggling through his third round of edits from our co-authors, so you can commiserate with him if you run into him. The other thing we did was looked at some of the relationships with the zooplankton as well, and I think that this is pretty common sense, that a lot of this is all zooplankton size. So a lot of zooplankton is smaller in systems that have alewives come into them. But you know, it's just not a very significant change. And one of the things that we've sort of come out of this work is that actually you see a big change if the fish are landlocked, because they graze as adults and juveniles their entire life cycles, so they're really graze down these large-bodied zooplankton. In lakes with anadromous runs, it's a much more nuanced thing, whereas the populations decline later in the year, you seem to get a recovery of a lot of these large-bodied zooplankton. And we've actually, thank you, Karin, for the paper on bridging the energy gap, we really think actually a lot of predation is occurring from the run itself. So you get this influx of tens of thousands or hundreds of thousands of adult river herring, and they are just grazing down the zooplankton. I don't know if they're preparing the zooplankton for their young offspring to come later, or if it's just a competition, and that's something that we have an undergraduate student working on. It's rather interesting, though. So just the summary, we see a lot of condition impacts that seem to be translated into a difference in growth rates of these predators in different systems. Less clear impacts on the zooplankton. We think that there's a much stronger driver of zooplankton biomass and sizes and species composition dictated by landlocked populations compared to these anadromous populations. I spent a little bit of time in the ocean. I'm going to be honest, we really know nothing about their ocean movements. We have some limited information that suggests that maybe they spend a lot of time near shore during their first couple of years of growth. But that's because we're sampling there really intensively, and so it's not surprising to me that we see them there. But the question is, are they elsewhere? We don't really know that. And so I'm going to treat the whole ocean as like a big black box, like I said it was at the beginning of the talk. And this is some mass balance modeling that my graduate student, Bia Diaz, has been doing. So this is the North Atlantic ecosystem in one slide. And this is one of those slides. It's like multiple years of work. And so what we have is from sources, detritus and phytoplankton, to where the target of the energy is. In this case, there's some copepods. There's some menhaden. Here's alocines, which includes all of the river herrings. So that's both blueback alewife and shad as well, because you really, in these map models, you can't just break everything up in the way you like it. You have to kind of go with what you have good data for. Here are the top predators. So you have some pelagic sharks. There's the top predators, humans, with all of their gear-removing fish. This is all based on the year 2000. And this is the trophic level up here. So what she did is she created two mass balance models initially, and asked the question, what would happen if you added back in this number of fish? Okay, so now we all of a sudden have a reconnected ecosystems. What happens? Well, here is the biomasses of the different groups under both restored, so that's restored connectivity, and contemporary, the current situation. And the thing you're going to notice, first of all, I noticed that she had this bigger bomb in her last draft of this manuscript, but you know, that happens, right? So in the restored systems, there is a lot more biomass. But the biomass is not just this Holocene group, which is right here, not a particularly large group. It gets spread out across a lot of other things. And the reason is that you're creating another method of bringing energy from the base of the food web up through the system to the predators. So that reduces foraging pressure on a lot of the other forage fish in the Gulf of Maine. It's really just sand lance and Atlantic herring. And so those populations, sand lance we have no data on, so you don't even include them in these kind of models. Here are, where are those Atlantic herrings? So Atlantic herring sees a big bump in their biomass. And that's just because they are not being consumed as heavily as they had been when they were the only thing that could be eaten. And so this work has really suggested to us that having a multi-species composition in your forage base in a dynamic and heavily migratory system like an ocean is kind of important. And we spend a lot of time sort of teasing out, because the other thing is that there's other species here like menhaden that are also not particularly abundant across this range, and sort of losing this multiple different prey species that these predators can consume is having some impacts on taking the energy that the ocean is producing at the base of the food chain and then putting it on our plates or for our recreation. And I'll just say that what Bia is doing right now, and she's probably actually doing it right now, is running scenarios in EcoSim, looking at what happens if you change the amount of fishing or increase the number of dam removals. What is like the way to get forward that's reasonable? Because we're not just going to get rid of all the dams and make this totally restored system. That's pretty unrealistic. Okay, the next part of this talk is all about timing, but also about just sort of starting to fill some of these data gaps. I'll try and link it back to the forage base as much as I can, but just recognize that having two species interact with each other is sort of like meeting your spouse. You know, you just have to be in the right place at the right time. I don't mean to compare predation to marriage, but anyway, I just did. So here is a lamprey that's, or even worse, a lamprey stuck on a gill arch of an alewife. But this required well, it required a bunch of things to happen. It required the lamprey to find the alewife in the ocean, then for us to catch the alewife at the ladder, and then for us to put it in a bucket, which we just happened to put a camera in and watch this thing happen. So I'm actually amazed that it stayed on the entire time. All right, quick little aside, we're experiencing warming. Everybody agrees with that, right? We saw a nice figure showing it. All right, so over that warming, there's a seasonal trend, obviously, as you go from summer to winter, summer to winter. And what we do a lot of times is we try to intercept at some point along this line just to understand. So it could be a migratory species, it could be growth rates going from warm to cold weather, anything that oscillates. Here's our little migratory species going in and out from north to south. So we get a lot of potential movements and things that are driven by these by this oscillation that's going on, but also with this increasing climate. So often what we'll do is we'll take a transition period. So, and these transitions that we've been using are defined by, in particular, the spring one, as the moment that the date by which the temperatures warming in the spring have crossed the average temperature for the entire time series. Okay. And so over time, what you'll see is that this goes earlier and earlier if it keeps warming, and the summers get shorter, the winters get longer. All right, in the river herring, our transition for this is going to be when 5% of the run has passed a counting station. This is work that Becky Dalton's doing in collaboration with Michelle Stogginger. We've also got a downscaled flow model, which I'm not going to talk about today because this work is really just not quite finished yet. So these are the systems we worked on. They have a whole bunch of different characteristics to them, including numbers of dams and number of fishways, all of that stuff we're trying to account for in the models. I was just, this is one of those things, like people say, oh, the run of river herring in the spring is temperature driven. And I'm like, well, where's the paper that says that? It's the spring, of course it's temperature driven, but what is the thing that actually drives the run? I want to know what drives that run. And so we spent a lot of time looking. Temperature actually, as it turns out, does not drive the run at all. It seems like if anything it's flow. Here what we have is the spring transition and the run initiation. So this is the only time that you pick up a temperature signal at all, is just on the initiation. And it seems to be related to this broad scale transition date, not the actual temperature itself. It doesn't seem to play much role. A little bit of variation, right? Most of this variation, it appears, is driven by the flow rates. All right, so and it's nonlinear, of course, just to make it really simple to model. So this is work that's ongoing, but if the timing of the arrival is changing, that sets up the potential for there being all kinds of trophic dynamic changes, but also changes in what happens when they get into the systems. I almost saved the methods just for the end, but what we have here, good. So what we have here is our pelagic purseines. We do this. This is how we sample river herring. In these coastal ponds, the river herring are actually not very available to this gear during the day, so we do all our work at night. It's really fun. I've learned that as my research has gone on, that all of my sampling seems to be happening at night, and I'm not going to say it's the greatest thing ever. But anyway, there's increased detectability, and actually what you see is the fish are, everybody watch Aliens, where you see the aliens coming down the hallway, coming to you, and well, that's kind of what river herring, although not anywhere near as scary, that's what kind of river herring do in these ponds. You actually see them move off the bottom as it shifts to night, and so they all of a sudden become available to this gear in the middle of the night, actually right around dusk. So we do this, we repeat our sampling over time. There are random sampling locations throughout the ponds. We've actually changed this. It's actually, we've done some work simulating how many toes we need. We actually needed a little bit more than we started with. So now we do 5 to 15 over one or two nights. It adds up really quick. We do an awful lot of sets per summer. We do all kinds of things, preserve otoliths, preserve fish for otoliths and gut content analysis. We also do some dry ice and other things to save those. Then we work up a lot of fish for their otoliths to get ages and also increment widths for daily estimations of growth rate. And then we collect as much as we can out of all of this work, including zooplankton, vertical tow. We did those multi-panel gill nets for predators, although we don't do that anymore because most lake associations are not big fans of gill nets. I wonder why. And then we have our temperature data, which really the temperature loggers are the most interesting part of this. So the first study that I'm just going to go over really quickly sort of follows up on like, okay, so the timing can change. It can be very variable from year to year. So what we did is we caught fish here at the Woolen Mill Dam. We fin clipped them, and this is Magna, our graduate student working on this project, and then we stocked them into Pentucket Pond. Then we went back in with our purseines and we collected them at night. And then we took all of this information and did a pedigree reconstruction of which adults produce which offspring. So this is a system that has not got a natural run, only fish that we stocked made it in. So you produce these things which basically show a yellow female and a red male, and these are the offspring they've produced. We stocked on a number of different dates, and so what you see is you can just sort of see that the fish are mating to produce these offspring that we are recognizing. And of course, it just gets really complicated. If I showed you the whole thing, it's just this mess of spaghetti lines. So you can sort of look at individuals and say, oh, so here's female. She was added, female 35, that's a really great name, was added, but she spawned with fish that were added weeks later. And so, and this is consistent. We see this all the time. You look at any fish, even if it's a female that was added at the end, she spawned with males that were added right at the beginning. These fish are not leaving systems quickly. They are staying in them for over a month, at least. The cool part about this work, though, is what she did is she then estimated the impact of the total length of the adults we added, and looked at the number of offspring that were assigned, or whether an offspring was assigned. So this is just now a logistic regression, and estimated the impact of the total length on these, on the likelihood of having an offspring. Really strong effective length, really strong effective transfer date. Early fish produce more offspring. I mean, it kind of makes sense, right? You have longer to produce more offspring. Fish that were earlier were also a little larger, so you have to sort of take it both into account. But the basic story is if you get in early on day one, you have a 20% increased chance of producing offspring. We then followed that up with some work where we extracted otoliths out of these fish, small little things, aged them, figured out how many days they'd been, took the age histogram and turned it into an estimated spawning time based on the temperature of these lakes, and then compared it to that count data. And what we saw was that there's just a separation between the first fish that go into the end of the pond. We're not seeing their offspring. So it's good to be early, but we're not seeing them. Well, we think they're just getting eaten. We think there's a lot of predation on these eggs and larvae right at the beginning. And it's not just one pond. Here's a stock pond with 4 different dates. It's every pond. Every pond shows the same thing, a big delay, and also our estimated spawning goes on for often a month after the fish have left the system, and there's no more monitoring at the counting station. So this is changing the way that we're sort of perceiving these dynamics. We thought the adult fish weren't playing a role in the ecosystem dynamics. Well, it looks like they probably are playing quite a role. We've continued this on to look at another suite of systems from Connecticut all the way up to Maine. This is another great summer of work. Sort of showing their growth increases over time, but we see just huge differences in different ponds. This is our stock pond, Pentucket, and this is a natural run, Gilbert Stewart in Rhode Island. You know, double the size of these fish at the end of time. Well, done some modeling on it, and it turns out it's juvenile density that's really driving this. Although, boy, it's complicated because they're playing a role in the zooplankton biomass, the adult run is playing a role in it, and so we're just still, this is another thing that's just in the middle of, I'm sure Matt's working on it right now, actually. These are our stock sites. So our stock sites are always lower abundance. So we know that there's a really strong effect of density, but this from year to year, we just see also big swings. There's a lot more going on here than this. The differences in growth, so if you look at these top couple ones, which I'm not going to expect you, but these are our stock sites compared to some of these other sites. This differences in growth start right away on like day one, basically, and they propagate through time. If you look at it, the growth relation to temperature, so temperature is this line here in the pond, you can see that it's tracking the temperature really closely, but it's not perfect. And so one of the things we've been looking at is sort of this 25-degree threshold. We think that there's something happening at 25 degrees that seems to impede further growth. So we have a graduate student, Leon Gao, who's now doing some lab-based studies looking at different ration levels and different temperatures. Take-home message: high ration, 21 degrees does best. You double the ration in 25 degrees, you don't achieve that same level of growth. So systems that have large numbers of individuals getting in there, those fish will remain smaller and available to predators for a longer time period, but they'll also drag down the zooplankton community a lot as well. And so there's a lot of really complex interactions for systems that I thought were going to be simple to work on when I started. All right, so there's a lot of moratoria. They're being reconsidered now for listing under the Endangered Species Act. There's a lot of reason to start knowing more about these fish. I was quite surprised, actually, in my first interactions with folks over this species, really poorly understood in terms of their dynamics in these coastal systems. You can't figure out solutions if you don't know what the problem is, and we really just are missing so much information, including like what's an optimal run size? Is it like the biggest you possibly can get? We don't think so. We think that there's probably, I mean, you're all fisheries biologists, you've all heard of density dependency, so what is the optimal size of a run? And it's going to be different for different systems, and probably, unfortunately, different from year to year as the nutrient levels fluctuate and other things happen. So we have a long ways to go in sort of illuminating these black boxes, but we're slowly picking away, and we're working on a really interesting project with the Mass Division of Marine Fisheries, where Gary Nelson, who's just an outstanding modeler, is now modeling the life history and how energetics relates to the growth rates, which relate to the immigration of fish. Then he applies different productivity, different systems, and can ask what kind of impacts could we have from offshore bycatch as these fish are moving through the oceans. And we feel like with that as a basis and with this data that we're collecting, that we might finally get to a place where we start understanding this fish and really helping to make conditions right for recovery. And the recovery is important. We control like probably this part of it. Maybe a little bit this one, these fisheries interactions, but they're really tough because they overlap with everything. It's like you go out there trying to catch an Atlantic herring, you're going to catch a river herring. But they feed a bunch of stuff, and they are interfering with our ability to have whales, because we see that if you increase the river herring abundance, you increase the potential productivity of these cetaceans. We just know that they're playing a really important role. And with that, I will say thank you.